Bismillahirrahmanirrahim. This is the video about molecular basis of cancer. This is the first video on this video series, which will describe the cell cycle from normal towards oncogenesis. These are the references. The historical perspective for the pathogenesis of cancer. For centuries, the cancer was considered as a mysterious act of the god, and in 18th century in London, John Hill established the cause and effect relationship between tobacco use and cancer. He also defined a criteria to define this cause and effect relationship, and that is termed as Hill's criteria. In 1775, Percival Pott associated scrotal cancer with soot in chimney sweeps. In 19th century, the bladder cancer was associated with alanine dye in Germany. Patenross in 1911, he established that avian cancer is by a filtrable agent, that is a virus. By 1920s, the human exposure to X-ray via fluoroscopy led to cancer. In 1941, Barenblum proposed a two-step theory of chemical carcinogenesis, and 1953 was the era which changed the things, and Watson and Crick identified DNA as the genetic material of the cell and elucidated the structure of the DNA. By 1971, A.G. Knudsen, he described that the cancer is due to involvement of two mutated alleles of RB gene in the development of retinoblastoma, and these are the tumor suppressor genes. In 1974, genes responsible for defective DNA repair in the skin disease, that is xeroderma pigmentosum, was linked to visceral cancer, and in 1976, Bishop and Wormus, they demonstrated mammalian genetic homologs that are called proto-oncogenes of viral transforming genes. When mutated, these cellular genes may become growth promoting gene that can lead to cancer formation. There are protective mechanisms in this cell that control the development of the tumor due to dysfunction or dysregulation of the gene expression. These protective mechanisms are cell cycle regulation, DNA repair mechanism, and telomerase activity. When these protective mechanisms are dysregulated or disturbed, the genetic aberration may lead to the development of formation or of a tumor. As described by Gregor Mendel, the gene is the basic physical and functional unit of the heredity. Now there are areas in genome that were considered non-coding earlier, and they proved to produce new classes of RNA later, and these RNA influence the gene expression. So the definition of the gene is now a sequence of nucleotide in DNA or RNA that encodes the synthesis of a gene product, either RNA or a protein. While the mutation, as we know that the cell division is not perfect, and there is some change, some aberration, and some difference from the original that is very negligible, and that is regulated by the protective mechanisms that are the DNA repair mechanisms and other protective mechanisms. But what is a mutation? The variation in DNA sequence may result from germline changes or acquired alteration in somatic DNA as a result of single nucleotide substitution or insertion or deletion of one or more nucleotides is termed as mutation, while polymorphism is variation in DNA sequence that are not associated with a known disease. The phases of cell cycle are quiescent stage, that is G0 stage, and from G0 the cell is propelled to the G1 phase, and from the G1 phase there is S phase, that is synthetic phase. G phase is growth phase 1, and the synthesis phase is the phase in which the DNA synthesis occurs, and following DNA synthesis the cell goes to the growth 2 stage and ultimately to the mitosis stage. If this cell is a cell of variety that replicates continuously, such as the intestinal mucosa and hematopoietic progenitor cell, it remains in M phase and it repeatedly replicates. And if it is present in the G0 phase, such as the hepatocytes that replicate infrequently, there is some stimulus that propels this cell from G0 to G1 phase, and then the cycle follows.
The cyclins are protein which control the progression of cell cycle by activating kinases that are cyclin dependent kinases. Cyclins were first discovered by Timothy Hunt in 1982, whose hobby was cycling, so he named his discovery on his hobby. When the cell receives stimulus to divide by growth factor, these cyclins are activated and they form dimeric complexes of two cyclin dependent kinases or CDKs, that are 4 and 6. These CDKs are important in the inactivation of retinoblastoma protein. With cyclin E, the cyclin D push the cell to the synthetic phase, and cyclin A is present throughout the phase from synthetic to the early mitotic phase, while cyclin B is present throughout the mitotic phase. The cyclin A and B bind with cyclin dependent kinases 1 and 2.
The regulation of cyclin and cyclin dependent kinases is important to protect against the development of tumor. When a cell receives a signal by a growth factor and cytokines, it is activated and there is increased gene expression that promotes cell division and increased proto-oncogenesis. This will lead to the action of early phase cyclins. The cyclin D and E, they activate cyclin dependent kinases 2, 4 and 6, while cyclin A activates cyclin dependent kinase 2 and 1, while cyclin B activates cyclin dependent kinase 1. Due to activation of these cyclin dependent kinases, there is formation of maturation promoting factors. These maturation promoting factors, they activate other proteins through phosphorylation. If there is a DNA damage, error in DNA replication, or some oxidant injury to the cell, this activity of cyclin dependent kinases and cyclin is inhibited. This can occur by the action of TP53 directly or indirectly by the activation of cyclin kinase inhibitors. The cyclin kinase inhibitors are INC, CIP, KIP and RB families. The activity of cyclin-dependent kinases require activation by cyclins. Therefore, its activity is limited by the availability of the cyclin. These cyclins are synthesized only when needed. They perform their function, then ubiquitinated and degraded by ubiquitinase protease system. The cyclin-dependent kinase inhibitor or CKIs, they strongly inhibit cell proliferation and collectively represent an important protective mechanism against oncogenesis, as we have seen earlier in the picture. They inhibit the cyclin by binary Cyclin-CDK complex binding or binding with CDKs directly. Hence, they stop their function. These cyclin-dependent kinase inhibitors are mutated in oncogenesis. The important cyclin-dependent kinase inhibitors or CKIs are INC4 protein, CIP, KIP and RB. The INC4 binds and inhibits CDK4 and 6. It inhibits the CDK4, therefore it is termed as inhibitor of CDK4 INC4 and prevent them from forming complexes with cyclin D. Therefore, it blocks cell cycle in the G1 phase, where the cyclin D was pushing or propelling the cell towards the S phase. Okay, so in G1 phase it stops the cell cycle. There are several inhibitors of CDK4 that are P15, P16, P18 and P19. The P14 ARF is encoded by same genes as P16 INC4A but is an alternate reading frame. Therefore, it is termed as P14 ARF, alternate reading frame. These proteins play important role in the regulation of both CDKs and MDM2. Other group in cyclin-dependent kinase inhibitor family is CIP-KIP, and this protein binds and strongly blocks CDK2 and to a lesser extent CDK1. They take up where the INC4 family, inhibitor of CDK4 family, lives off and inhibit the rest of the cell cycle. The important member of CIP-KIP family are P21, P27 and P57. Among these, the best understood is P21. The RB protein is another family of CDK inhibitors. It also targets CDK2, and expression of cyclin-dependent kinase inhibitors can be induced by senescence, contact inhibition, extracellular anti-mitogenic factors, for example, transforming growth factor beta and the tumor suppressor protein P53. Beside these cyclin-dependent kinases inhibitors or CKIs, there are other mechanisms of inhibition of cyclin and cyclin-dependent kinase activities. One of these is selective post-translational modification, in which there is phosphorylation of some of the cyclin-dependent kinases and their inactivation. Another mechanism is that there is
presence of a specific nuclear export system that prevent the cyclin and cyclin dependent kinase dimers accumulation within the nucleus, which is their site of activity. So the proliferation of the cell is inhibited. This is the summary of the events that we have studied up till now.
The retinoblastoma protein and its family also regulate the cell cycle progression. They act near the G1 to S phase boundary. The prototype is P105RB, which is simply termed as PRB. The PRB inhibit progression of cell cycle by binding with the members of E2F family of transcription factor, hence these are inactive. These E2F factor mediate cell entry into transit through S phase by increasing transcription of downstream cyclin A and E as well as other protein that promote DNA replication. Binding of this E2F by RB blocks the E2F activity. Phosphorylation of RB by complex of cyclin D, CDK4 and 6, they alter RB conformation and release E2F factor. E2F mediated upregulation of other cyclin A and E which complexed to CDK2 again phosphorylate RB, allow the cell cycle to progress. The RB gains back its inhibitory function as the cell division completes. During anaphase, PP1, that is protein phosphatase 1, dephosphorylate RB, and this RB binds back to the E2F. Hence, it stops further division. There are two landmark transitions in the cell cycle. One is G1 to S, that occurs when the activity of the complexes that involve cyclin A and E, they overwhelm inhibition by CIP and KIP family of inhibitors, while G2M phase transition occurs when cyclin B and cyclin dependent kinases 1 complexes are activated by removing inhibitory phosphorylation of CDK1. During the cell cycle proliferation, there are some checkpoints which stop this cell progression if the need arise. These checkpoints are four. One is the one checkpoint is before the entry of cell cycle into the synthesis phase, another is during the synthesis phase, and then there are two more checkpoints that are during the G2 and M phase. The checkpoint 1 and 3, that is before the start of S phase and that is during the G2 phase, are activated by DNA damage and hence they stop the cellular proliferation, while the checkpoint 2 and 4 are activated by other factors. When there is a damage to DNA by some physical factor, or if the DNA replication is defective, there is activation of these ataxia, telangiectasia mutated and ataxia, telangiectasia mutated and RAD3 related kinases activation. These kinases activation activates or phosphorylate the P53, and P53 is released from its inhibitor, that is MDM2. The phosphorylated or activated P53 then activates P21. The DNA damage, if it is in G1 phase, the P53 directly increases production of CIP and KIP cyclin dependent kinase inhibitors, while if this damage is in G2 phase, it can occur. The stoppage of the cell cycle can occur by two mechanisms. One is the p53 mediated downregulation of cyclin D and cyclin dependent kinases 1, or there is activation of two related enzymes that are checkpoint kinases 1 and 2 that block cell cycle progression immediately. The restriction point is during G1 phase. It is the point where cell decides whether to proceed with mitosis or not. This restriction point is activated by cyclin D, cyclin dependent kinase 4 and 6 phosphorylation of RB protein, and there is subsequent release of E2F to facilitate mitosis. Once this R point is achieved, external factors affecting mitosis has got no role now on the proliferation, and this proliferation is only by intracellular mechanism. The loss of this restriction point control occurs in many cancer and dysregulate progression through the cell cycle. There is another checkpoint, that is replication checkpoint. When there is a exposure to a toxic agent and the cell become idols in S phase, the nature activates check1 kinase that blocks mitotic separation of incompletely duplicated chromosome. During mitosis, each cell should receive correct complement of chromosome. This is achieved by activation of spindle integrity checkpoint that sense the correctness of division. It is activated by sensors at the kinetochores, that is the area where chromosome attaches to the mitotic spindle. There is another enzyme activation, that is Aurora B kinase enzyme, and this activation ensures that the effectiveness of the binding of the kinetochore and effectiveness of the mitotic spindle. P53 is termed as guardian of the genome, and it coordinates cellular response to DNA damage. It mediates activation of G1 by S and G2 by M checkpoint, and it initiates apoptosis. The P53 is maintained at low level by binding with the MDM2, that is murine double minute, and it is a E3 ubiquitin ligase that binds this P53 to ubiquitin and triggers its proteasomal degradation. The damage to the DNA is sensed by ATM and ATR. The ATM is ataxia telangiectasia mutated, and ataxia telangiectasia mutated and RAT3 related is ATR, and they recognize this DNA damage, and in combination with check1 and check2 kinases, they phosphorylate P53, which usually is present in combination with murine double minute, and when this is phosphorylated, this P53 is released from the its complex and activated, and activation of this P53 through these mechanisms or via telomere dysfunction contribute to cell cycle arrest. Now this checkpoint activation has a purpose. It is giving time to repair the DNA. When this P53 is phosphorylated, it is dissociated from the murin double minute and it is translocated to the nucleus. In the nucleus, it further promotes the cell cycle arrest by stimulating P21. P21 inhibits the cyclin action on RB phosphorylation. So there will be arrest of the cell cycle. Simultaneously, this P53 also activate GAT45, which is responsible for the DNA repair. If DNA is repaired, the block is removed and the cell cycle proceeds. And if the process fails to repair the DNA, the BEX is activated and this leads to apoptosis. Mutation in cell cycle gene will result in development of tumor. Any regulatory molecule gene may be affected, such as cyclin, cyclin-dependent kinases, cyclin-dependent kinases inhibitors, and RB gene. And their mutation disturb the regulatory function, and many antineoplastic drugs, they target these genes to limit cellular proliferation. So this is summarizing all the events we just discussed. The P53 remains in low concentration within the cell and it binds with murin double minute, which is E3 ligase, and it binds it with the ubiquitin and leads to its degradation. The genotoxic stress, which is recognized by check 1 and check 2 and ATR and ATM, will result in phosphorylation of the P53, and P53 phosphorylation will transfer this into the nucleus, where it activates the P21, and this activation of P21 will result in the arrest of the cell cycle by stopping the action of cyclin on the RB. So RB is not phosphorylated, so the cell cycle is arrested. Simultaneously, the P53 also activates GAT45 that results in DNA repair. If DNA is repaired, the cell cycle continues, and if it is not repaired, the P53 will activate BEX gene, and BEX gene activation will result in apoptosis. The human body is made of billions of cells. These cells are under continuous stress by environmental chemicals, ultraviolet and ionizing radiation, reactive oxygen species, intracellular milieu that break down DNA chemical bonds, infidelity of the DNA polymerase, which may result in single or double stranded breaks within the DNA, single nucleotide substitution, base insertion, base deletion of variable lengths. There are certain cellular vigilance mechanisms that collaborate among diverse proteins and restore this genomic integrity after such DNA modification, as these DNA modifications are not intended to convey or to carry into the daughter cell. So the cell repair itself by these cellular vigilance mechanism, the DNA is repaired, or if the cellular mechanisms they fail to repair these mechanisms, there is activation of apoptosis. This illustration is describing the DNA alteration that results from the stress, various kind of stress, and their repair process that is associated with that alteration. There may be a single strand break that is repaired by base excision repair, or a double strand break that is repaired by double strand break repair. These can be non-homologous end joining or homologous recombination. If this repair process fails, the representative tumor type is pancreas, breast, and ovarian tumors. The DNA adducts are formed and they are repaired in nucleotide excision repair, and the failure of this repair mechanism of nucleotide excision repair is exemplified by xeroderma pigmentosum. Base insertion or deletion and base mismatch are repaired by mismatch repair, and the example of the representative tumor is colon and rectal tumors. The mismatch repair pathway is mainly involved in repairing errors in DNA replication. The error rate in replication of the genome are different. These are for somatic cell, there is one miscopied base per 10 raised to the power 9 bases. For the germ cell it is less. It is one miscopied base in 10 raised to the power 11, and for polymerase delta and epsilon the error rate is 1 base per 10 raised to the power 4 to 5. The difference between these error rates of the polymerase editing and the final product largely reflects the effectiveness of this mismatch repair process. There are two mechanisms of this mismatch repair system. One is the correction of single base mismatches, and another is fixation of insertion and deletion that is caused by slippage during DNA replication. The mismatch repair system is the cellular vigilance system that is activated when there is mismatch or insertion or deletion. When there is mismatch from the original sequence during DNA replication, that is recognized by MSH2 and MSH6, and the mismatch repair 1 mediators are activated that repair this defect and the original sequence is restored. While the insertion or deletion is recognized by MSH2 and MSH3, and the set of the mediators that are activated are MMR2, and these MMR2 mediators, they repair the defect that occurred in the DNA and they restore the original sequence. If the enzymes of this MSH system that stop the DNA replication at G2M checkpoint, they are unable to repair the defect of the DNA, they initiate apoptosis. The defective mismatch repair system can be inherited, or this can be defected through acquired mutation that develop over time in somatic cells, or through epigenetic silencing of a component of mismatch repair system. An important indicator of this defective system is microsatellite instability. Microsatellites are short sequence of up to 6 base pairs that may be repeated as many as 100 times. These are common in human genome and prone to mutation, including the number of repeats can be changed, and that result in the mutation. Microsatellite mutations are detected and repaired by mismatch repair system. If escape repair in germline or somatic cell, these mutations may indicate the likelihood of cancer development. An example of this is colon and rectal carcinoma. Nucleotide excision and repair mechanism is another mechanism that is the cellular vigilance mechanism which correct distortion of DNA helical structure, and it identifies and corrects the bulky DNA adducts, base oxidation, reactive oxygen species from the mitochondria and other sources, the damage by ultraviolet radiation, and the damage by ionizing radiation. This nucleotide excision repair system repairs the DNA by two mechanisms. One, it is constantly scan the genome. This mechanism is inherently deficient in xeroderma pigmentosum, while other mechanism of the repair of the DNA in this nucleotide excision and repair system is the identification of alterations that interfere with the RNA transcription, and this mechanism is defective in cocaine syndrome, that is also an inherited condition. If any of this mechanism finds a defect in the DNA, it repairs both of these mechanisms. They repair the defects by the same mechanism, that is through an enzyme, that is endonuclease repair enzyme, ERCC1 enzyme. This enzyme is very important and it has got practical therapeutic importance, because as we already know that the tumor cell, they tend to repair its DNA to protect them from the chemotherapeutic agent that is targeting its DNA, the tumor cell DNA. So the sensitivity of the tumor cell to ERCC1 will help to predict the sensitivity of the cancer to the chemotherapy, whether this particular tumor will respond to that chemotherapeutic agent or not. The base excision repair is another cell surveillance mechanism that is related to the nucleotide excision repair mechanism, and these two mechanisms overlap in the type of DNA damage to which they are responsible. The base excision repair mostly repair chemical injury to DNA bases such as hydrolysis of base sugar bond, single strand breaks, and small chemical changes in base structure. That alterations occur about 10,000 times per cell per day, and these insults are usually caused by environmental and other chemicals, reactive oxygen species, ultraviolet radiation, and ionizing radiation. The inherited defects for base excision repair are not described. Another mechanism of cellular surveillance is double-stranded break repair. The double-stranded break can be caused by reactive oxygen species or ionizing radiation or stalled DNA replication even after a single strand break. The sensors of this double-stranded DNA break are ATM and ATR enzymes. When this double strand break is due to ionizing radiation, this is sensed by ATM enzyme and it immediately recruits CHK2 or CHEK2 enzyme, and if it is by the stalled DNA replication fork, it recruits CHK1 or CHEK1 enzyme that will lead to the phosphorylation of p53, and activation of this phosphorylation of the p53 will stop the progression of the cell cycle, and this DNA will not replicate unless this is repaired. So this double-stranded break repair can be non-homologous end joining and homologous combination. And the examples of these chromosomal rearrangements that happen in the tumor of pancreas, breast, and ovary. Homologous combination
is mostly active in S and G2 phase. The template which is used for homologous combination may be sister chromatid or homologous chromosome, and it may result in two type of genetic alteration. One is the loss of heterozygosity, because the allelic difference between the two chromosomes may be lost as the homologous chromosome is used. Then the homologous repair or homologous recombination repair of a break that happens to occur in repetitive sequences that occur frequently in human genome, and in one chromosome it may lead to use of an identical repetitive sequence of a non-homologous chromosome as a template of recombination. Thus, this generates a translocation. Several genes such as BRCA1, BRCA2, PAL-B2, whose protein products are important for the homologous recombination, are often mutated or inactivated in many tumors. This may occur as part of inherited cancer or may occur epigenetically during oncogenesis. The non-homologous end joining is another cell surveillance mechanism in which the break ends are directly ligated without the need for a homologous template. There is no guarantee that the ends that are so joined are in fact the broken ends of the same chromatid. That results in the development of translocations, and these non-homologous end joining mechanisms operate throughout the cell cycle, while the homologous end joining were mostly seen in S and M2 phase. They are more efficient than homologous repair, and they use Q protein, Q70 and Q80 proteins, and this mechanism is the mechanism that is involved behind the VDJ, the variation, diversity and joining recombination that is responsible for the generation of diversity in B lymphocyte, and it functions well and without any error, but it is not easily account for the original base order of the DNA sequence it joins. When there are multiple double-stranded breaks, this non-homologous end joining may link non-contiguous DNAs. The defective non-homologous end joining is not associated with any non-inherited predisposition to the developing cancer, or it is not commonly associated with oncogenesis. This table is summarizing the type of the damage, their causes, and the repair pathway that is activated in response to that damaging insult. The various type of damages are basic oxidation or additions, intra-strand DNA cross-linkage formation, single-strand nicks, or double-stranded breaks, as we have seen earlier. The mitochondrial and reactive oxygen species, phagocytosis, generated oxidants, ultraviolet and ionizing radiation, and smoking, they activate the nucleotide excision repair and base excision repair pathways, while the other base modifications which result due to chemotherapeutic drugs, neutrophil bactericidal enzymes, cigarette smoking, and other environmental chemicals, they activate base excision repair pathway. The alteration perturbing the DNA architecture, one is the addition that is caused by the errors in transcription, reactive oxidant species, and ultraviolet light, and they activate the nucleotide excision repair pathway. Inter-strand DNA cross-linkage are caused by ionizing radiation, arrested DNA replication, and environmental chemical that activate other mechanism such as Fanconi related repair pathway. That is another mechanism of this repair. The single-stranded nicks are caused by reactive oxygen species, ionizing radiation, spontaneous loss of sugar phosphate bonding, and they activate the base excision repair and nucleotide excision repair pathway. And double-stranded breaks are due to arrested DNA replication, ionizing radiation, and chemical damage, and they activate homologous recombination and non-homologous end joining pathways.
There is a definitive role of genetic and epigenetic alterations in the development of cancer. This conclusion is made possible due to advanced DNA sequencing and other technologies. Some genetic themes have evolved due to these advancements in reading the body's building element. These themes are that the non-lethal genetic damage lies at the heart of carcinogenesis. A tumor is formed by the clonal expansion of a single precursor cell that has incurred genetic damage. 4 classes of genes are the principal target of cancer-causing mutations. Carcinogenesis results from the accumulation of complementary mutations in a stepwise fashion over time. Mutations that contribute to acquisition of the cancer hallmark are referred to as driver mutation. Loss of function mutations in genes that maintain genomic integrity appears to be a common early step on the road to malignancy. Mutation in many other genes contribute to the tumorigenesis by interfering with host immune responses. The non-lethal genetic damage lies at the heart of carcinogenesis, means that there is genetic damage but it is not resulting in the death of the cell. The environmental exposure to some element, it may be inherited in the germline, or it may be spontaneous and random, that will result in initial damage or mutation. These insulting agents may be exogenous or endogenous. The exogenous agents are viruses and environmental chemicals, while the endogenous agents are the product of cellular metabolism that have the potential to damage DNA, such as reactive oxygen species, or they alter gene expression through epigenetic mechanism. Some of them are termed as oncometabolites. Another genetic theme is that tumors are clonal. A tumor is formed by the clonal expansion of a single precursor cell that has incurred the genetic damage. A normal cell, if it has got a mutation, it is transformed into a precursor cell, and then there is proliferation of this precursor cell, and all of these daughter cells, they contain the initially acquired mutation. These mutations are identified by DNA sequencing, which is helpful in identifying the point mutation, or these are also studied through chromosomal analysis, which is best for chromosomal translocations and copy number changes. 4 classes of genes are the principal target of cancer-causing mutations. These are growth-promoting proto-oncogenes, growth-inhibiting tumor suppressor genes, genes that regulate apoptosis, and genes that are responsible for DNA repair. When the growth-promoting proto-oncogenes are activated, there may be one or more cellular functional increase that are encoded by a gene that promote tumorigenesis, or the appearance of a completely new function, that is oncogenesis. The carcinogenesis may result from accumulation of complementary mutation in a stepwise fashion over time. As is evident by this picture, a normal cell receives cancer-inducing mutation, and by this carcinogenic inducing mutation, the cell is transformed into a precursor cell that has got stem cell like properties. With the addition of further driver mutations, a cancer cell is formed, and with additional mutation and further genetic evaluation, the tumor may form subclones, and it is now genetically heterogeneous at the time of at the time of diagnosis. The driver mutations are the mutation that contribute to the acquisition of cancer hallmark. The initiating mutation is the first driver mutation that start a cell to the path of malignancy. This mutation is maintained in all the daughter cells of the subsequent cancer, but the cancer is developed after acquisition of more driver mutation because no single mutation appears to be fully transforming. The timeline to gain this mutation is unknown. One example is childhood acute lymphoblastic leukemia cell. They bear initiating mutation that is seen in blood sample taken decades before the diagnosis. The persistence of initiated cell is consistent with the idea that these cancers arise from cells with stem cell like properties that have a capacity to self-renew and long-term persistence. So they are termed as cancer stem cells. Function mutation in the genes that maintain genomic integrity is another genetic theme, and when this genomic instability is introduced, there will be increased likelihood of acquiring driver mutation and increase in the frequency of mutation that have no phenotypic consequence. These mutations are termed as passenger mutation. These passenger mutations are more common than the driver mutations. Then another theme is the mutation in many other genes contribute to the tumorigenesis by interfering with host immune response or altering interaction with the stroma or by other mechanisms. Once established, the tumor will follow the Darwinian selection or the survival of the fittest theory. When there is a driver mutation in a cell, and this is a clone formation by this driver mutation, and there may be some apoptosis of the cells that forms a clone of the tumor cells. Then this tumor progresses and gains further driver mutation, and this acquisition of the additional mutation is random. So the tumor at the time of diagnosis may show genetic heterogeneity, and the tumor cells must be about 1 billion to get clinical attention, and 1 billion cell means it's about 1 gram of the mass, and 1 billion cells formed after 30 cell doublings. So this is how the tumor is evolved, and these diverse clones of the tumor, they compete for access to the nutrients and microenvironmental niche, and those clones or those subsets that are most fittest, they win this struggle and come to dominate the tumor mass.
Tumor progression is the tendency of the tumor to become more aggressive over time.
Besides genetic mutation, the epigenetic aberration also contribute to the malignant properties of a cancer cell. These are the DNA methylation and modification of the histone. DNA methylation result in silencing of a gene, and as a result the function is lost, such as if the tumor suppressor gene is silenced, it will result in the development of cancer. Then there is a modification of histone, and the histones are the proteins that are responsible for packaging of the chromatin, and their alteration may result in overexpression or underexpression of the gene expression.